Good morning. Hi, Arvind.
How are you doing?
So today we'll be talking about virus evolution. I'm really excited to learn about the work that you do right now, your research. Can you tell me a little bit about your background?
Yeah, my identity is quite complicated. I'm from Kenya. I'm born and raised in Kenya, but my parents are of Indian origin. So the British moved a lot of people to Africa as part of their colonies. I've moved around the world. I did my undergraduate in England, and then I moved down to South Africa, where I did my PhD, and I had a faculty position there. And then I moved to New Zealand for about 9 years. I came out to ASU, say, 4 years ago.
Wow. What do you study?
So as a molecular virologist, I study viruses. And the word molecular is pretty broad in its own way. It's anything that is within a cellular kind of entity. And in my case, I study a lot of the viral evolution and how the viruses behave when they encounter new hosts, also including discovery of new viruses, because we keep finding new viruses in different systems. And I focus more on environmental angles. So I do a lot of work within ecosystems rather than mainly focused on, say, human health. When you start thinking about emergence of viruses like outbreaks, if you don't know what's in the ecosystem, you have no idea what's going to come out. They're all part of this big picture.
Can you talk a little bit about what is virus evolution?
So virus evolution in general is how do viruses evolve? What are the mechanisms by which they evolve? And one thing that we need to remember when we think about evolution of viruses compared to evolution of, say, larger mammals like us, is that our life expectancy is different. So, what happens with a virus infection, you're getting through one cell cycle. In some cases, let's say bacteria, which replicate every 20 minutes. Within 20 minutes, it is producing, in some cases, up to 10,000 offsprings. So you can imagine when you're making that many offsprings, you can have a variety of different mutations. So you've got a lot of variation that comes out of one replication cycle. So, you've got variety of different ways that viruses evolve, and one of them is mutations. The other is when you get co-infection taking place, when you've got two viruses entering the same cell, but they're closely related to each other. And then what they can do is when they're replicating, they can swap templates. They start making chimeras, as we know about the chimera, half human, half horse.
Right.
You can imagine how easy it is to then make completely different genetic elements very quickly.
Yes.
And then you've got a third mechanism, which is very unique, which is some viruses, their genomes are segmented. They're fragments like chromosomes. So most viruses have one chromosome as their genome, but other viruses have multiple. So imagine you've got two viruses coming into a cell with these multiple chromosomes, which each encode different genes. Then you can get mixing taking place. So just think about, most people know what M&Ms are, and they come in different colors. So you separate them by color. You just have the red ones and the green ones, and you throw them in a bowl, and someone asks you to pick 8 random ones without looking. It's highly likely that you will have a mixed coloration of what you pick out of the 8. And that's exactly what happens in some of these viruses. And a beautiful example of that is influenza virus. That is why every year we have to get revaccinated, because they're changing constantly.
And you actually study beneficial viruses as well, right?
Yeah, we kind of try to.
Okay.
So I think the way we as humans have looked at viruses or studied viruses for the past 50 or so years is very much from a very biased angle, primarily because they cause disease. With modern medicine and human health, animal health, plant health, everyone's been looking at things that they really care about. And when they see that it is sick, we start studying it in greater detail. And so a lot of our virus knowledge is very much biased towards those that cause disease. And there is very little that we know about in terms of what they do in ecosystems. You talk to the marine biologists, and they'll tell you viruses are very important in ocean systems because they infect different organisms at different trophic levels. And then on terrestrial systems, there are some beautiful examples, and one of the best ones that is in Yellowstone National Park. There is a grass that grows at about 55 degrees centigrade, which is pretty warm for anything to grow. Even us to stay in that temperature for a whole day, it's not easy. But there is really, really beautiful biology that takes place there, and that is that the grass is infected by a fungus. But the beauty is that fungus is infected by a virus, and that virus basically modulates the growth rate of the fungus. So what happens is if you take the virus out, the fungus really grows rapidly and kills the grass. And you take the fungus out and the grass dies. The fungus somehow activates certain genes in the grass that allows for it to be tolerant to these high temperatures. And these three coexist.
Wow.
In this beautiful relationship.
So for the grass, the best situation is to be infected with a fungus that has the virus, right? If the fungus doesn't have the virus, virus will kill the grass?
Exactly.
That's fantastic. That's such a beautiful example.
So, Dr. Varsani studies evolution in really fascinating places, from the hot Sonoran Desert in Arizona to the frozen continent. Here, we talked about his work in Antarctica.
What kind of work do you do there?
So in the Antarctic, I'm doing work on a variety of animals there, mainly penguins. And the beauty of the Antarctic is that it is probably the only continent where there has been the least amount of human activity. And so with that, if you want to study from an ecological context viruses and how they interact with different organisms that live there, perhaps that's one of the best places. One of the other things is also important is that that area is facing a lot of climate change. Animals are struggling for breeding sites because it's a lot warmer than they usually breed at. And so with that, the animals are likely to be a lot more stressed. And with stress, we know in our systems, we as humans, when we're stressed, we're more likely to have infections come up.
Right.
And the classical example is cold sores.
Yeah.
We keep it under control, but as soon as we're stressed, we start seeing it pop up. So similarly, these animals, when they're stressed, chances are they'll be more prone to pathogens. And so we're trying to see what these dynamics are.
And I think that a lot of people don't realize that we carry a lot of viruses.
Exactly. And a lot of it doesn't infect us. It just passes through our gut. If we think about plants, we eat a lot of plant material.
Right.
Your salads, etc. Some of it, might have viral things in there. Basically, it goes through our system, doesn't infect us at all. If you look at most individuals' skin, we'll have papillomaviruses on our skin. You rarely see any kind of manifestation of disease in some of these things. Mm-hmm. So we do know a lot of viruses have co-evolved with humans. Mm-hmm. And so sometimes when we change their conditions, do they become a little bit more detrimental?
I have so many questions. Quick one, because we're talking about Antarctica. There are several horror movies that suggest that if some kind of an ancient virus was melted and it would spread, it would just destroy the whole population because we wouldn't have immunity to it. Is that at all possible, or is that crazy Hollywood?
Well, it is possible, but it's not as crazy as that. Okay. The thing is, if that virus does come out, for viruses to infect and get into a cell, they need to interact with what's on the outside of our cells. So if those, we call them receptors, so if they can't attach to them, so it's almost like if you can't fit into the shoes, you can't go out for a run. Simple as that. If they can't attach to it, they can't get in. And also, once they get in, they have to adapt to the system to see whether they can use that system. You can be a tractor mechanic and you can try repair a scooter, but your tools are going to be different. And so it's the same thing. You might have come infecting something, but you have to readjust and make everything ready. And by that time, immune systems would have kicked in. So don't forget that we've got a pretty major line of defense. We've got a first line of defense and a second line of defense. Between those two, we should be able to manage it. It is one of the things that people worry about, especially in Siberia, where a lot of individuals who were buried in some of the ice, which is now melting, they were actually buried in an era when they was infected with smallpox. And you've got grave diggers and bounty hunters that are just going around trying to see if there is anything available that is of ornamental value. They might get infected if it has been preserved really well. Possibility does exist, but we haven't seen a case of it so far.
I wanted to ask you, I believe you do some science communication and you have a penguin costume.
Yes, I do. I have many penguin costumes. So I think it's kind of important for scientists to engage with the rest of people who are part of the community. There's a lot of jargon and complex words that we use. One point, if we really are going to do all this research, and if we just write it in the sophisticated languages, 3/4 of the community don't understand it, then what is the point? And so I think it becomes very important for us to actually articulate our science. With that, the community will appreciate, number one, some of the findings that we have, will be much more believing of science.
Yeah.
Because in a lot of instances, people just see this jargon and it's just a shutter has gone on. That's it. Because what we're then doing is we're creating a class system. And with class systems, you basically get to a level where people just switch off.
Yeah.
And like, okay, that's that class and this is our class. And we'll deal with this in our own way. If anything, we're just marginalizing people.
And Arvind, what would you be interested to learn more about?
I would really be interested to learn a lot more about the black hole. And I think that is a very, very unique thing to try and distill down. It's not easy. And that's something that I'd love to see in a very, very more simplified context.
That's something that's just so hard to wrap your mind around because it's a bit out of this world. Arvind, thank you so much for your time. And I hope to see more of your science communication.
Thanks a lot. Take care.